Log on, tune in, find out. Another good idea from Cambridge.
Today it's my pleasure to welcome our speaker who's come a very long way, indeed from Auckland Cancer Society Research Centre in New Zealand. That's Graeme Finlay, where he's also been working doing research for the past 20 years. Graeme is also senior lecturer in pathology in Auckland University. Now, in parallel with his work on topoisomerase 2, for those who like those sort of things, but particularly in cancer therapeutics and the like, Graeme has also developed a great interest in comparative genomics over the past years. And that's what led him to write these books and indeed led to his invitation here this afternoon. And it's this latter interest in particular which is the focus for our theme for this evening. And we're very pleased to welcome Graeme, and he's going to talk to us on human genetics and the image of God. Thank you, Graeme.
Thank you, Dennis, and thank you everybody for giving up your evening to come to this lecture. I hope at the end of it that you consider it's an hour well spent. Thank you also to CIS and the Templeton the Faraday Institute for your very kind invitation to bring me out here. I feel very much a boy from the bottom of the South Pacific coming to this awesome place, Cambridge. And I thought perhaps as I start I should just mention the different conditions which have led me to the particular interests which Dennis has already referred to. On the one hand, living as a Christian in New Zealand, I'm very aware of the incredible controversy that tends to rage in circles there regarding man's place in the biological scheme of things and the theory of evolution. Unfortunately, it's a very hot topic there, and so much of our theology comes from America. It's almost a Christian orthodoxy to believe in a young earth. And that's concerned me greatly over the years. On the other hand, we're also very aware that there are people at the other pole of thought who like to insist that if you believe in evolution and that the evidence for evolution is very strong, that thereby displaces any reason for believing in God. And Professor Dawkins' book hit the bookstands just as I arrived in the country. And so we really need to address this issue and wonder why there is so much controversy over it. The second reason is, as Dennis has said, my background in cancer research. And it is the incredible advances in cancer genetics which have led me into considering evolutionary genetics over the last 20 years or so. And my interest in cancer genetics was greatly fanned in the early 80s as the story of retroviruses and of oncogenes grabbed the headlines and radically changed our understanding of the molecular basis of cancer. Oncogenes are a fascinating set of genes, retroviruses are a fascinating set of viruses, of oncogenic viruses, because their genetic material is something called RNA, and it is read backwards or reverse transcribed into DNA, which is the more standard genetic material. And there's just an image of some cells with lots of retroviruses, in fact the human T-cell leukemia virus type 1 budding from it. And human T-cell leukemia virus was discovered in this era, in the early 80s, as the first retrovirus which was oncogenic in human beings. And this diagram here, wrong light, this represents the virus with the genetic material inside and the proteins outside. Now, the life cycle of human T-cell leukemia virus or other retroviruses occurs something like this. The infecting virus fuses with cells because it's got a sticky protein on the outside membrane. its single-stranded RNA is then injected into cells, and then using this fancy enzyme, the reverse transcriptase, the RNA is transcribed or reverse transcribed into double-stranded DNA, which is the more orthodox form of genetic material. And then the retrovirus does something very sneaky. It inserts its own little tiny genome, its own little segment of genetic material into the genetic material of the host cell. So there is a particular insertion point which generates small duplications on either side, which we call target site duplications. And for any one retrovirus, that tends to be unique, because the retrovirus can insert into multiple places, myriad places, in an infected cell. And so we have a situation like this, where here are, say, 3 infected cells. This dark bar represents the DNA. The red arrow represents 3 different sites where the retrovirus inserts itself into the host genome. And thereafter, the retrovirus is simply carried from generation to generation as long as the cell lives and divides. So it's a consummately clever parasite. And in its location as part of the genetic material of the cell, it can then generate copies of itself in perpetuity. Now, the fascinating thing is that if one of these insertions produces a tumor, such that, say, 40 years later, a patient comes down with a human T-cell leukemia, what you find is that in that leukemia, which might have 1,000 million cells, you find the retrovirus genome inserted at exactly the same place in every leukemia cell. Why should that be? Why should the retrovirus insert randomly at multiple sites, myriad sites, through the host genome in the infected stage? But later, when there's a cancer, the retrovirus is there at one particular place. There is no way that it can insert with fidelity, multiply at the same time. Its mechanisms don't allow that. The reason is that the impulse to develop into a cancer arose in one particular unique cell with its unique retroviral insert. And that one cell thereby started to divide and form an expanding clone. And then with other genetic events, many years later, produced a huge clone of descendants of perhaps thousands or billions, thousands of millions of cells, all of which received the one particular provirus, that endogenous retroviral insert from the one precursor. And here's just one particular example taken from the old literature. This is a study looking at mice. Up here we have a small segment of a chromosome. And for those of you who aren't biologists, the A's, the T's, the G's and the C's represent the 4 letters, the 4 chemical letters which constitute our genomes. I have blocked in here a little area in yellow which represent the target site of an incoming retroviral genome. And that target site is GTTT-GC. That is the target site of an incoming virus. When the virus goes in, this pink patch represents the virus. It's actually 9,000 A's, G's, T's and C's long. But we haven't got room to put all that. So I've just put in just a few with all the dots in the middle. Here is the target site duplication, which indicates the way, the mechanism, the tried and true mechanism by which retroviruses insert. This original insert would have been a unique event. But when the tumour appears, every tumour cell will have this retrovirus inserted at exactly this site with a GTTGC target site duplication. Now, what's this got to do with human evolution? All this has been worked out with infective retroviruses. But as people started to study genomes, they made an amazing discovery that there are retroviruses in our genomes which we have received not by infection, but by inheritance. You and I contain something like 100,000 retroviruses, retroviral genomes, in our DNA. And the 100,000 retroviruses which I contain are identical, pretty much, to the set of 100,000 retroviruses which you contain. And in fact, there are related types of parasites in our DNA, LTR elements, totaling with the retroviruses 400,000 in common. So you and I contain the same 400,000 retroviral elements, inserts in our DNA. Regardless of our ethnicity, folks, we are 1/12 virus. Where do they come from? Our parents, obviously, and our great-parents and our great-grandparents. How far back do we need to go to find where these retrovirus inserts appeared? Sorry. Okay, here we go. We'll be down. Here is just a little segment of DNA from a human cell. And this red line here, sorry, Dennis, I think I'll need another. Oh, hang on, let's come back. That was me. That was you. Okay, now I'm really confused. Thanks. Cell biologists draw DNA off in this red lines. It's just on my day.
Press the red star at the top.
Sorry?
Press the red star. This one's working too. Yeah.
That's wrong. Okay, sorry. Anyhow, that black line, that black line represents a bit of DNA, perhaps about 10,000 bases long. And these other little bars I've drawn on it represent what we call restriction enzyme sites. Think of them as little milestones, which indicate some of the basic features of this DNA. So, thanks. I don't know. Right. Down below it is a clone, is a piece of DNA from a chimpanzee. And it's the comparable piece of DNA to the human clone DNA. So you can see that just about in every case, the chimpanzee's milestones are the same. So we are dealing here with the same piece of DNA. And below it at the bottom is one of these endogenous retroviruses. There is its end here, and Pol and Env are two of its genes. And that endogenous retrovirus is here, somewhere in this chimp DNA, and somewhere in this human DNA. And the implication is, because the milestones are so similar, because we are dealing with essentially an unaltered segment of DNA in human and chimp, that retrovirus will be the same. The implications were dramatic. This is early 1980s. If it could be demonstrated that we and the chimps had at the same site, with the same target site duplications, the same piece of retroviral DNA, then ineluctably we would conclude, just as in the tumour situation I mentioned to start with, we had received that endogenous retrovirus from a common ancestor. Sorry, what's happening again? Okay, so the story would be something like this. Now we are talking about germ cells, reproductive cells. And in this case, retroviruses can infect germ cells. Many of these will be lost from history. But every now and then a retrovirus might infect a germ cell, which is passed on to future generations, and then to multiple future generations, and then potentially to new species. There are 3 species of ape. And they would have the retrovirus, what we call the endogenous retrovirus, present at the same site. And by analogy with everything I know about cancer cell biology, this would be to a biologist formal proof of common descent. Now I know a physicist and a philosopher would take great exception to that, so I'll retract that statement. The first study to actually look at the DNA, to look at the sequence level, came about 10 years later. And this looked at 3 endogenous retroviruses, these 3 up here, which were found at the same site in humans, 2 species of chimps, and in gorillas. And the conclusion is that these retroviruses must have integrated in a common ancestor of the humans, that's a new species, humans, chimps, bonobos, and gorillas. 3 other endogenous retroviruses were found also in gorillas, orangutans, gibbons, and in as many species of old world monkeys as you care to study. That red bar shows the distribution of those endogenous retroviruses, and the conclusion is they must have integrated into the primate germline in a common ancestor of all these species. And here is just one example. There is an endogenous retrovirus, 9,000 bases of it, and I'm not showing those. There is the target site duplication, and it's present in humans, chimps, bonobos, and gorillas, although part of it's not given. The uninterrupted target site is present in the orangutan. There is a target site, but there's no there's no endogenous retrovirus, there's no target site duplication, and with orangutan the sequence simply reads TGAGACAATAT without any interruption. And I have some other examples as well. There's another one linking humans, chimps, and bonobos. And there's another one linking humans, chimps, gorillas, and this time orangutans. It's the gibbon, which has the uninterrupted target site. Dear folks, I hate controversy, but when I came across this data, I felt I had to do something, because the entire fundamentalist campaign against evolution can be shown to be demonstrably wrong. When we look at this data, we can see that there is absolutely no doubt possible that we have common ancestry with other primates. And if Christianity, if the Christian faith which I profess is not based on truth, then it sinks to just another postmodern story. And so this is why I entered into the controversy and tried to start to tell people that there was no longer any need, any possibility of valid controversy about the past evolution of the human species. Well, since that time, the human genome has been sequenced, all 3,000 million base pairs of it. And then in 2005, the chimpanzee genome was sequenced, all 3,000 million base pairs of it. And here we find the number of endogenous retroviruses which are held in common with human beings and with chimps, and it comes to over 400,000. 400,000 in common. Same target site duplications, same element. There are a few which are human-specific, type 1 and type 2 ERVs, 5 of that and about 100 of that, and a handful which are chimp-specific. Clearly, these are ones which have inserted subsequently to the common ancestor, and they indeed show all the properties that you would expect of relatively recent insertions. They have accumulated far fewer mutations because they've only been in our DNA for a few times, for a relatively short time. And in fact, if you look at these human-specific insertions, some of them are polymorphic. Dimorphic. In other words, some of us in this room will have them, some of us in this room will not have them. So in other words, they are so recent that they haven't spread to the whole human population. So here is overpowering evidence of common descent, which we cannot deny. In New Zealand, where I come from, where there is this tragic controversy, we hear that evolution is just a story that adults, a fairy tale that adults believe, or there is no evidence for macroevolution. Maybe people don't worry about that in Cambridge, but I just appeal to you that these rumours, these falsehoods which are spread by certain uninformed branches of the Christian faith, be resisted resolutely. And I say that without rancour and without anger, but it's a tragic thing. Well, let's just continue. We have other denizens of the genome as well, which can add to this story. There are related elements which cut and paste themselves around our DNA. Our DNA is a very, very active environment. And these are called retrotransposons. Retro again because they copy themselves backwards through an RNA intermediate. They start off with an original element, original parasitic piece of DNA sitting safely somewhere on a human chromosome. They make an RNA copy. That RNA copy travels somewhere else in the genome. It makes associated enzymes which produce a staggered cut in target DNA, just like these retroviruses. And then a DNA copy is made with target site duplications, usually with target site duplications. And then the breaks are filled in, and you've cut and pasted a new element in our DNA. And with every 20 new babies, another one of these appears in the human germline. So it's ongoing. We are still accumulating these parasites. And they constitute, together with endogenous retroviruses, 50% of our DNA. 50% of our DNA has been added stepwise by the copying and pasting activity of these elements. And here's just one for example. This is called an L1 element, line 1 element, and PA7 is just the family up here. And it's present, you can see the left flank and the right flank are exactly the same except for a small deletion in a selection of the new Old World monkeys. But it's the same element in humans, chimps, gorillas, orangutans, and Old World monkeys, establishing common ancestry. And systematic studies, with the advent of widespread genomic sequencing, systematic studies have now produced the same, exactly the same family tree as you get from a study of the distribution of endogenous retroviruses. These are called ALU inserts, up there at the top. And here are different families which have been studied in the primates. And just looking at the pink box, that's a particular family in this particular study produced by a remarkable laboratory in America, the laboratory of Mark Batzer. Here in this study, 21 have been identified which are present in human beings. Here are 2 particular elements which are shared by humans, chimps, bonobos, and no other species. Here are 12 elements which are shared by humans, chimps, bonobos, and gorillas, but by no other species. Here are 20 elements which were shared by the great apes, but not by the lesser apes or the Old World monkeys. And here are 49 elements which are shared by all the apes, but not by Old World monkeys. So here, again, is staggering independent evidence for a common descent. The same studies, I'll just pass over these quickly, have been done by this group in the Old World monkeys. That's the monkeys of the African and Asian continents. And the numbers just represent the total number of elements which have been mapped in these studies and which are common by all the species listed. So there are 35 elements studied which are common to all of the Old World monkeys. 33 elements which are common to these, 13 elements which are common to those, etc. The same study has been done with the New World monkeys of Latin America. And the same approach has also been used to resolve long-standing mysteries. Let's go down to the very, very base of the primate family tree. And we can ask, we've got anthropoid primates such as us, how do we relate to the tarsiers, which are just a small group of prosimians, and the lorises and lemurs? Well, three possibilities have been suggested by different scholars, different primatologists at different times. How do you resolve the issue of who is a sister group to who else and which group branched off first? Was it the lorises and lemurs branched off first, the anthropoid primates branched off first, or the tarsiers branched off first? And this mystery has proved wholly tractable by the use of ancient alu elements, 4 of which have been mapped in the DNA of anthropoid primates and tarsiers, but are absent from the DNA of lorises and lemurs. Once again, cogent evidence of common descent of anthropoid primates and tarsiers right back at the base of the primate family tree. And just for completion, these elements have also been used to delineate the precise relationships of the lorises, lemurs, and bush babies, the galagos. So in summary then, just some cute pictures. I'm a cell biologist. I don't think about whole animals. Small things amuse small minds, and I'm normally satisfied with cells. But here are some of the wonderful little creatures which we now know to be our biological relatives. And also a sense of sorrow that we are quietly wiping most or many of them to extinction by cutting down their forests. So to summarise this section, we have this enormous catalogue of ancient insertions in our DNA, all of which mean that our DNA is an astonishing history book. It's not only a blueprint for our biology, but it's an astonishing history book of our past history. And when we look at the line 1 insertions, of which there are 800,000 in our DNA, and comparing the human and chimp genomes, 99%, more than 99% are shared. There are a few families, 2,000 recent ones in human, 2,000 recent ones in chimp. And also the other group, the alu elements, 1 million are shared, 7,000 are in human beings, 2,000 are in chimp, and here are 2 other ancient elements. The mirrors are particularly interesting, exceedingly ancient. We share those with other mammals. And that's another story in itself. And essentially all are, of course, shared with chimps. So folks, our DNA has been put together stepwise by the action of parasitic elements, which are still jumping around. But not only are they adding themselves to our DNA, but they are contributing to our very biology. These jumping genes are contributing some of the genes by which we grow and live as human beings. And I just want to give you a couple of quick examples. We've got a gene, for example, from a very ancient endogenous retrovirus, ERV, called WE1. That's the endogenous virus, that's the target site duplication, and here it's present in right down to the gibbons. Old World monkeys aren't shown, but it's present in them as well, but not in New World monkeys, prosimians, or the dog. Right, now this endogenous retrovirus has contributed to our biology one of its genes called ENV. ENV is the virus gene, the endogenous virus gene, ubiquitously present in endogenous viruses, which enables the virus to stick to cells as an early event in the infection process. And normally when a virus is endogenized, when it becomes part of DNA, it just starts to collect mutations and wither away. It just starts to merge into the genetic background. But ERV1 has an ENV gene which is still intact in our DNA and that of chimps, gorillas, orangs and gibbons after 40 million years of residence. So it still makes an active protein. And that protein is still functioning. If you take that protein from any of these species, from the DNA of any of these species, and insert it into cells and culture, that protein is still sticky. It can be expressed on the surface of cells and make cells stick together, make them fuse to form what is called a syncytium. And current theory has it that the normal expression, these are experimental conditions, but the normal expression of this viral protein forms a syncytiotrophoblaст, which is an essential part of the developing early embryo. And so, a bit like Pip finding his benefactor was Magwitch, the runaway criminal, we find that our benefactor in many cases may be viruses, which have contributed genes to our very biology. And that is just the shape of the syncytin-2 gene, which is what the name of this endogenous viral gene is. Here it is, it's a spiral, couple of 3 big alpha helices in it, and it pretty much exactly overlaps a current endogenous, that's a mouse Maloney-Neuron leukemia virus, ENV protein, and the current human T-cell leukemia virus, ENV protein. So it still retains its viral shape after 40 million years in our genome. And just very briefly, this is an example of convergent evolution. The Conway-Morris hypothesis of convergence. And here is just a very recent paper, came out just before I left Auckland. We have the syncytin 1 and 2 genes, I've talked about syncytin 2, totally different viruses, endogenous viruses, have contributed other genes which do the same function in the reproduction of mice. And the Yacht-seeked endogenous retrovirus has contributed exactly the same function in the reproduction of the sheep. And in the sheep it's been shown that if you suppress the production of the endogenous retrovirus, the embryo dies. So there you are, but our very lives are dependent upon an endogenous retroviral insertion 40 million years ago. Now, I think maybe I could just briefly mention that endogenous retroviruses can sometimes cause other unique random events in our DNA. Here's a gene. Those 3 red blocks represent sequences of a gene which are made into RNA, which are processed into a messenger RNA and used to make a protein. Sometimes the enzymes made available through these parasites can associate with the RNA and insert that RNA back into the DNA of the cell to form what we call a copied retrogene. Most of these are junk. They don't do much that we know of, although that may be speaking too early. But sometimes they can produce new active genes. We could call it irreducible complexity arising de novo. There we go, there's one of them. The GLUD2 gene is one of these reverse transcribed processed genes. There it is, I haven't put the sequence in. There are the target site duplications which indicate that it arose as a result of the fortuitous association of parasite enzymes with a cellular RNA. And there are a whole lot more. I've just listed some there. We won't go into them now. And again, we see convergent evolution. Here's one called UTP14, and it has been inserted back by this random process at least 5 times in independent events in mammalian evolution? Well, we've learnt a lot about our genomes. I think this is a fascinating story, and it establishes beyond reasonable doubt that we are continuous with the rest of the primate order. And this raises a question. Now we know so much about our DNA. That's the royal we. I'm not a genome person. I'm just an onlooker. Can we track down the genes that make us human? And this has been described as the critical question now of anthropology. And you see these sorts of cliches. These are two headings from Nature of relatively recent times. We're homing in the genes for humanity. It's a very grandiose thing to say. Are we really homing in on the genes for humanity? Well, the gene hunters have got a lot of work on their hands. Because even though we are so similar to chimps, with 1% difference in our A's, G's, T's and C's, nevertheless, that 1% is still 17.5 million bases. We differ from them in 2.5 million insertions and deletions. We have 180 duplicated genes which chimps don't have, 160 retrogenes, 90 inactivated genes, and we have 70 to 80% of our genes make non-identical proteins. So when we see this rather facile statement, we're closing in on the genes that make us human, don't be misled. They are definitely scientifically frameable questions. But it will be a very big and long task to find out what makes us human. And in fact, one wonders whether the totality of our biology will arise from the totality of our interacting genetics, and that the reductionists are going to have a very hard time indeed, looking for specific genes. But can we go on a little further? And here I was very stimulated by the recent book by Professor Lewis Wolpert, 6 Impossible Things Before Breakfast. And in this book, Professor Wolpert has, as a good reductionist, he calls himself a materialist reductionist atheist, whether he is also a fundamentalist is another question, he writes this, that there may also be genes for religion. Do we contain in our DNA, as part of what makes us uniquely human, genes for religion? And Professor Wolpert writes that religious beliefs may be genetically programmed into our brains. In other words, he says, are there circuits in our brain that are set up by the genes that predispose us to have religious and mystical beliefs? Well, this is a very interesting question, and I think it's an interesting question to answer, to consider just briefly over the next few minutes. First of all, let us assume that what Professor Wolpert says is entirely true. That in fact religious beliefs may be in some way genetically programmed in our brain. Hang on, is this my hand shaking or what? Thank you. If Professor Wolpert were completely true, if the God of the genes hypothesis is true, I believe that it is still fully compatible with the idea that God ordained that it should be there. For example, let us think about rationality. I'm sure Professor Wolpert would insist that rationality is something which was naturally selected during the development of human beings. But just because it was naturally selected does not thereby mean that it has no traction on reality. And if rationality has traction on reality, then I think it is fair to allow that religious belief also has traction on reality, no matter how it was selected for during our evolution. What about another example, parental care? I am quite happy to allow that my passion for my children, these feelings of deep love and empathy I have for my children, were naturally selected during evolution. And that in a consequentialist sense, it is good to care for my children. But that is wholly compatible with a religious perspective which sees that in a deontological sense. it is right, it is morally right to compare, to care for my children, because that accords with the moral law of the universe, which reflects the nature of God himself. A consequentialist evolutionary ethic is in this case fully compatible with a deontological religious ethic. And thirdly, I think St. Augustine may agree with Walpert's hypothesis, because St. Augustine said, man's instinct, Lord, is to praise you. You have made us for yourself, and our spirits find no rest until they find their rest in you. Augustine said man's instinct, this deep, primeval urge, if you like, is to respond to God and to adore him. And therefore, an instinct arises from genetics and the way we are, and so I have no problem with this. But I would like to question a couple of areas of the Walpert hypothesis, because firstly, I think he needs a slightly higher degree of exactitude in his thinking. One thing he says when he's comparing, when he stops talking about science, is to compare the marvels. Stops talking about religion, is to compare the marvels of science. And he says science is not genetically determined. As if to put it on the pedestal, as if to compare it with this crude animal instinct by which people turn to God. But I would just like to suggest that when he compares science and religion, science or religion, he is comparing dissimilar entities, dissimilar features of the human experience. The equivalent of science, yes, chalk and cheese, sorry. The equivalent of science is, thank you, sorry, Becky, is theology. It is science and theology which are not genetically encoded. It is science and theology which arise from the work of trained scholars who have been inculcated in the disciplines and the methodologies of venerable traditions. It is science and theology which proceed by the communal activity of scholars working according to a common canon of procedure. And I would suggest that the equivalent of religion, which may be causal beliefs about God in Walpert's terms, is in fact causal beliefs about the physical world. I would suggest that it is causal beliefs, the ability to entertain causal beliefs, whether of a religious nature or a scientific nature, which is genetically encoded. It is this which defines us as human. The ability to ask questions about God and the ability to ask questions about cells or evolution or history or atoms. Those together, I would see as part of our genetic endowment, part of what makes us human. Thanks, Dave. And finally, I would say that theology and science and the underlying genetically encoded impulses which they give expression to reflect on history. As a Christian, I would say God's action in human history, in personal history, and God's action in physical and biological history. And therefore, I think as we read 6 Impossible Things Before Breakfast, we should be very cautious about the Walpert hypothesis, because it divides human experience rather superficially. But I think the Walpert hypothesis also has another question that needs asking. Genes are not the whole story. Folks, we must insist that our biology is the result of genetic change, which we can follow right back. But genes are not the whole story. Experience or knowing molds brains, And the classical example, of course, is what happens in a baby if both of its eyes do not work. If a baby is born with a squint or with a cataract, that squint or that cataract must be repaired very quickly, because there is only a set period in the development of the brain when the neural system is tuned by the activity of the impulses from the eyes. And if that cataract is removed too late, it does not avail the child at all, because the neural tuning system has shut down, and the baby will never use that eye to its fullness. Our brains develop not just through genetic activity, but through experience. And there are many examples of this. I'm not a neurologist, but functional MRI has shown that musicians develop different parts of their brain, different structures, or different structures develop differently in musicians from non-musicians. Thanks, Becky. If you're a juggler, there you are, a recent paper in Nature. You develop different parts of your brain. The very morphology of your brain changes, not in response merely to genetics, but to the time you spend on your juggling. Thanks. If you're a London taxi driver, you've probably seen this study. You have differences in your posterior hippocampi, which a non-taxi driver doesn't have. And just to forestall the questions when they come at question time, the changes in the hippocampi are correlated with a number of years of taxi driving. So they are a consequence, not a cause, of being a taxi driver. And finally, if you're a suffering medical student applying yourself to major exams, then again there are parts of your brain which develop in a few months, which can be detected by this very exact science of functional MRI. So genes are not the whole story. And we have to allow that with human beings, genes and experience make us who we are. I'd like to quote an American neurologist who wrote this very provocative, very stimulating article in Brain and Mind. He says, "The development of socio-emotional functioning, early life nurturing, appears to be critical. If this is absent from the first 3 years of life, and then a child is adopted and begins to receive attention, love and nurturing, these positive experiences may not be sufficient to overcome the malorganization of the neural system, mediating socio-emotional functioning." Folks, genes are not adequate in the making fully of a human being. And I've taken that photo of a Romanian orphanage from the net. There are 60,000 of these children in Romanian orphanages who are growing up deprived of what we would consider to be essential and necessary parental love. 600,000 children in Russian orphanages. Thanks, Becky. And here is a picture in Perry's review of a section of an X-ray section of an MRI section of a normal child's brain and a child suffering extreme neglect. And of course, this isn't a controlled experiment. We don't know what effect in utero alcohol exposure or lack of nutrition had on the lack of growth. But I think it is general understanding that the lack of parental nurture and care has at least a part to play in the development of brain circumference in these children. And thanks, Becky. And here is a study that Perry also reports. The light grey bars represent the relative brain size of children at the particular age, 1, 2, 3 or 5 years, when they were removed from institutions where they were not given adequate love and care. And they are all there below, at or below, the 5th percentile of brain circumferences. And the dark bars represent the growth in brain circumference 1 year after being transferred into a home where they were accepted with love and compassion and care. Now, again, these are not controlled experiments, but it is known that if you do the experiments with marmosets or with rats, you feed them all they want. You don't expose them to alcohol. Yet boring environments produce boring brains. Stimulating environments produce stimulating brains. So we have to conclude that nurture, such a gossamer thing, such an immaterial thing as parental nurture, is part of the necessary growth environment of the physical substance of the brain. Such a tangible reality as life, as the fullness of human life, requires such an apparently intangible thing as communication, nurture, love, communication, and words. And it reminds me of the words of Jesus Christ. Man shall not live by bread alone, but needs every word that God speaks. Here to our Western minds, and this is, I know, very easily criticized, but just accept to our Western minds, life is very tangible. We think in terms of molecules and cells and bodies. But life requires communication, nurture, care, and words. Such a tangible thing as life depends on such an intangible thing as words. And St. Peter, when Jesus asked his disciples whether they would leave him, he said, you have the words that give life. To me here is an amazing analogy of the role of communication, nurture, care and love in the very physical development of the human brain. And so we come finally to considering the real role of the gracious other in the development of humanity. It seems to me an amazing thing that the very development of our brains and minds requires spiritual qualities such as love, joy, peace, patience, kindness, goodness, gentleness, and self-control. How does the materialist cope with this? Our physical brains do not develop without spiritual qualities. Show me how you measure love and parental nurture, how you can quantitate it. And I will allow that there is truth to some of the claims of materialism. The forbidden experiment to bring children up apart from the nurture of a loving environment has in fact been done by Frederick II, the second of Hohenstaufen, almost 1,000 years ago. Becky, can you just switch it on, please? Yeah. And this is what he did. Bidding foster mothers and nurses, he wanted to bring up children apart from their parents, to see what language they would speak. Would they perhaps speak the language of heaven? Bidding foster mothers and nurses to suckle and bathe and wash the children, but in no wise to prattle or speak with them. For he would have learnt whether they would speak the Hebrew language, which had been the first, or Greek, or Latin, or Arabic, or perchance the tongue of their parents, of whom they had been born. But he laboured in vain, for the children could not live without clappings of the hands, and gestures, and gladness of countenance, and blandishments. This is recorded for us by the Augustinian Friar Salembini, who was a contemporary. It appeared that the experiment was surprising. The children not only failed to speak the language of heaven, but they died. They were suckled, they were nursed, they were fed, but without communication. They died. The genes for humanity did not kick in. The genes for religion were not expressed. Thanks, Becky. I think other analogies are the tragic stories of feral children. We don't know how many of these are purely apocryphal and how many are real. The story is told of two little Indian children who were discovered in a wolf lair. Perhaps they had been abandoned. and the mother had taken it home as fodder for her cubs, but perhaps the children started to suckle the wolf and maternal instincts took over. And these little children who were discovered by the Reverend Singh and taken home, they soon died. They did not learn to talk. They could not be socialized into society. In some deep and amazing way, they share our humanity. We know that. But they did not learn language, and perhaps not knowing language, they did not learn. They did not become self-conscious. Thanks. Macron, who's a neurologist writing in Lancet Neurology, says our humanity is the outcome of three kinds of adaptive history. The influence exerted by biological evolution through genes. That's wonderful, it's exciting, it's a sharp science, but it's not the whole story. There's the evolution of neural circuits through experience. The influence of cultural evolution. He says, it seems that culture and speech are essential for the makings of the human mind. It could be that the lack of a social trigger at the right moment meant that certain genetic programs failed to kick in. Thanks, Becky. Coming back to Steve's, he's a philosopher thinking about the phenomenon of the feral child. He says, as he thinks about how they fail to become socialized, perhaps they cannot learn language if they are found after a certain time. Perhaps they do not learn human consciousness. What I have in mind is the notion that the burgeoning consciousness of the infant will not necessarily develop into human intentionality on its own, but rather requires the presence of a significant other who is human. This gracious act of attention is thus responsible for creating a human person. Without a human other to attend to the child as human, the child does not become human. Which is not to say that feral children have no sense of self or other, but rather that such senses do not include humanity. Humans, we know, are not defined genetically or anthropologically. Humanity is in some respect the result of specific treatment within one's humanity, one's community. Being human is being treated by humans as human. Dear folks, as I stand before you now, I know, we are sure, that our genome has developed in specific increments from that of a human chimpanzee precursor. That is no longer questioned, but it cannot make us human. Our humanity, the fullness of our humanity, depends on such an intangible thing as human relationship. And I think there are such enormous implications here. I wonder what's happening to our children as they spend vast amounts of their childhood in front of a computer rather than with a household full of grandparents and aunts and uncles and the sick and the lame and the old and playing sport with children on the local green. What's happening in our shrinking families, in our communities with ever higher walls? The wonderful American theologian Stanley Hauerwas calls us a community of strangers. Our humanity requires community and interaction. And the genes for humanity and the genes for God or religion are impotent without that sort of interaction. And again we come to the words of Christ, words which for a long time were unclear, very vague to me. When in the Lord's Prayer in John 17, Jesus said, eternal life is knowing you, the only God, and knowing Jesus Christ, whom you sent. Such a concrete thing as life, in this case eternal life, which is embedded in our psychology, the life which is being integrated into the society of God. This eternal life comes from knowing, comes from interpersonal knowing. Eternal life is knowing you, the only true God, and knowing Jesus Christ, whom you sent. When I studied for my theology degree by correspondence with the University of South Africa, I learnt an amazing metaphor from one of the black tribes, the Xhosa people. A person becomes a person through persons. And if only we could learn in our molecular age of the reality of this wisdom. And if only we could see in the words of the Lord Jesus Christ that the fullness of knowing is the possibility of a relationship with God. I conclude with the words of St. Paul, which to me summarize the fullness of human life. Paul wrote, "All I want is to know Christ. All I want is to know Christ." Here is the knowing which creates us as people, to experience the power of his resurrection, to share in his sufferings, and become like him in his death, in the hope that I myself will be raised from death to life. And I thank you.
Well, thank you, Graeme, very much indeed for opening up a wonderful array of thoughts, and I'm sure that's also stimulated quite a few questions. We do have some time for questions. We won't go on very long, but we do have 5 or 10 minutes for questions before refreshments. If you have to slip away, there are exits at the back, but otherwise we'll stay here and go into discussion time. So who would like to ask the first question? There's a question here, yes.
Yes, thank you. I think early in your lecture you developed a very compelling case for the relationship between species. And I think speaking from the theological side, and I think the controversy, and really you've got to set aside the young earth creationists and that whole scheme, because there's no common ground there. But it seems that the controversy is actually more nuanced, which is the mechanism of moving from one species to the other. And it's not that the relationship is denied, I think, by the majority of Christians, but it's that mechanism which is not slow and gradual, but now is being called punctuated equilibrium and mutations or whatnot. But I was interested in the slide that you showed when you mentioned the question of what it means to be human and all the different genetic sequences. But if you compare that to the other species, what are the differences, and how do you move from one to the other? Not just showing that there's a relationship, but the actual mechanism which moves from one to the other, I think is the area of controversy really between the evolutionists and the Christians who are struggling with that.
Did everyone hear the question at the back, right at the back there? Everyone's okay? Right, good.
That's a very big question. And the only two things I can say is that as we look at these DNA sequences, all we can conclude are that these changes occurred stepwise and incrementally, one after the other. So clearly there must be a gradual change to transform one species into another. So we are talking about 5 million years between the chimp-human common ancestor and now. If your underlying question is, where does the image of God come in that gradualistic change? My short answer is, resoundingly, that I don't know. I can only suggest that we also look at our own development from one cell to responsible adult. And in that continuum ask, where do I become responsible? Where do I fully express the image of God? We don't know. So it is a question, I think, that no one knows. So I can't answer that, sorry.
Okay, so we'll have this question in the middle here. Yes? Yes.
Since we share so much of the genome, 99% with chimps, but that the genetic disposition is not everything that makes us human. Does it imply that chimps could be made human?
Now, I'm not a... This is getting a little bit out of my... a long way out of my area. No, it doesn't. I think chimps can be trained to do certain things, to represent, to make certain, understand certain words, to make certain sounds. But beyond a certain threshold, they can go no further. And I think because the interacting totality of our genome is so complex, they never will go further. So no, I don't think they can.
Yes, question up here.
What do you with St. Paul? St. Paul actually says that Christianity is not only unreasonable, but it is folly. That is to say, not only is it unreasonable, but it doesn't even pretend to be. Insofar as this, why do you feel the need for liberalism and science tools to justify the epistemology and the metaphysics of Christianity if we indeed, you and I both, worship this Jew with fourth grade education who happens to be our Messiah? That is to say, why do you feel the need to use the enemy's tools, per se, to disconstruct the enemy's house? Of course, that is not to say that what you have said today is not of tremendous import. We're hardly disengaged with Christians. But as a Harvard scholar and a PhD student in ethics, I feel that too much of what you're saying is probably engaging with liberalism's tools. Can you give us an answer to that?
The question is, am I using liberalism's tools today to defend Christianity? Is that the nub of it?
I feel like you're trying to justify, there's no need to justify what we do, because I feel that, this British time said, don't think but look. And I feel that Christianity has its own internal mechanism, its special privileged sacred language that transcends these justifications.
I agree with you. I mean, I haven't talked today about the basis of Christianity. I have just assumed it. It would require another sort of talk totally to look at the basis of the Christian faith. And I believe one of these is coming up in a Faraday lecture next year from, I think...
Bishop of Durham, that's right.
Right, also, and Dr. Tom Wright. Yes. So I was just... I mean, there are two areas which I really wanted to address. I just wanted to focus simply on the question, have we evolved? And I know that that those who deny it do represent an extreme group, but a very large extreme group. And so that's why I wanted to address that particular question, as well as to share the amazing biology which is being revealed. And the other, I do not consider I was using the tools of liberalism to talk about the essential importance of relationship, which is a very biblical concept, in fact, to the Jews, to the ancient Hebrews. Life was not a thing in itself, but rather an interacting network of relationship and knowing. So I don't think I was using the tools of liberalism in the second part of my talk by any means.
Okay, so that's a question down here. Yes.
I wonder if you could say exactly a little more about what you meant by spiritual values. You were talking about that in the context of loving human relationships, which of course is fine. But loving human relationships, of course, are not the characteristic only of religious people. And I wonder if you could say, what is the difference in your context between spiritual values and loving values?
Very little. I think love is something which is part of what makes us human. So loving values, nevertheless, in that context, I think they're synonymous.
In that case, the conclusion would be that all people who are loving should also be religious.
Well, that is partially true, but then I would just like to add the additional layer of love shown to us by God, which I think carries us beyond human love. That's the self-sacrificial, self-emptying love that God has shown. But I do not mean in any wise to belittle, say, the maternal love or the family love, which is the common property of all of us as human beings, and which is essential for the humanity of all of us. but which is not measurable in any materialistic, reductionistic sense.
Okay, yes, and red sweater, yes.
Incidentally, I think it was a simply splendid talk, if only to learn genetics. In your splendid talk, early on, you said that back home, where you come from, this is now an extremely divisive debate with a large number of people who are implacably opposed to accepting any evolutionary theory, and that you imagine that in Cambridge, perhaps in the UK, the situation is different. Have you any thoughts as to why this is so?
Or may I say, suggest why certain people should insist that an understanding of evolutionary mechanism eliminates considerations of purpose. The whole area, I believe, to be unnecessarily the area of controversy. I do not know.
Is it so back home at all, please?
What, that there are two polarities?
Why is creationism popular? Why is creationism?
Right. Well, I wonder if it is, one reason is perhaps because Christian teachers have failed to teach their flocks about the meaning of Holy Scripture. And much currently of the reason given for creationism is faithfulness to Scripture. But Genesis and other parts of the Bible properly understood reveal to us a God of faithfulness and goodness and rationality who comes to us in love. The Bible does not reveal science to us. And to look to the Bible for science is totally to miss the point of what it's making. So I think the bottom answer, or the bottom line to your question, is that there is a lack of theological education. I think there is another reason too, which I may perhaps just add, and that is that evolution has so often been taught as an alternative to religious faith. You know, I think America is the place of great confusion. But if you go back and look at the textbook that was used by John Scopes, and I've looked at it, I got it from the Auckland University Library, after very moderate sections on evolution, John Scopes' textbook identifies the Caucasian, as opposed to the black or the brown or the yellow human being, as the apex of evolution. Now that is there in black and white. If you look at the text, I can't remember its name offhand. That was John Scopes' textbook. And to me, William Jennings Bryan, the Christian who took John Scopes, or who took Scopes to trial, to me he was always an idiot. But when I looked at the textbook that he was trying to refute, he became a flawed hero, because he was probably rejecting the racism and the elitism implicit in early 20th century evolutionism. But unfortunately, he threw out the baby with the bathwater. And that to me explains so much of the confusion in America at present, which has been exported to so many countries. We must distinguish between various forms of evolutionism, in which science is used in the service of ideology. From Hitler onwards, you know, Hitler was supported by essentially all the anthropologists of his time, of his nation. We must compare evolutionism with the wonder of evolutionary biology, which is fully compatible with Christian faith.
Okay, I think we've got time just for one last quick question, and one right here, yes. Yep.
I was enormously impressed with your talk, and I felt it might have been helpful to have as a conclusion to assist your fundamentalist friends to start by looking at the fact that Christianity had to rewrite the creation story in order to have a coherent account that included the person of Jesus Christ in it. And it's most noticeably done in St. John's Gospel, which has to start in the beginning. And having done that rewriting of the creation story for Christians, then everything that you said at the end of your talk, where what matters is relational and requires words, is put into that prologue so beautifully that that's where I thought your talk could end. And I found that very, very moving.
I think that's a good note which to draw the discussion to close. But I'm sure you want to join me in thanking Graeme very much for a great talk. And